Okay, so again, thank you to those of you who are here today. We do have a good number of people who are absent, so hopefully they start to trickle in. That's why I was pushing back the time a little bit. I wanted to give some folks an opportunity to get here. But it looks like some people might not be joining us. They've started their weekend early or they have some technical difficulties. No big deal. But I just wanted to appreciate you all, say thank you for being here today. And most of you are here almost every day. So you're doing an excellent job with attendance. Let's keep it up. Keep it up. Hopefully that was enough time to do the warm up. Those warm ups are important, and you all have heard me say it every day this week, but I'm going to say it again to close out the week. Those warm ups are important because they give you an opportunity to see what the test questions will look like. They also give you an opportunity to review what we talked about in the previous day's lesson before we jump into some new material. So we always like to get that extra added review if and when possible. And now that we're about to jump into some new material, hopefully you already have your notebooks open because hopefully you were using your notes to do those warm-ups. I also want to remind you of one more thing. Those warm-ups are set so that you can take them an unlimited number of times. There is no limit on how many times you can take those warm-up quizzes. So I did that so that you all can do that in preparation for the exams. That's a good study mechanism to have. Go back and look at some of those test questions from the warm-ups and exit tickets because I take them from the exact same source that I take the test questions. So in many ways, they're very similar. And in some cases, you might find that they are the exact same questions that you'll end up getting on the test. So work smarter, not harder, folks. Use those warm-ups and exit tickets as study methods, even though you may have already done them. Go back and change some of your answers, look at the questions, think about what you may have learned that may help you answer the questions since you first did it. It's a great, great study tool to have. Today's lesson is all about genetic diversity. The title of today's lesson is Meiosis and Genetic Diversity. Today's date is February 5th, 2021, and again, it's the fourth day of our third unit. The objective today is to explain the role of meiosis in sexual reproduction and genetic variation. Same objective that we had yesterday, but today I'm adding one more. Biologists will be able to understand various sources of genetic variation. The essential question, how does meiosis ensure genetic diversity in sexually reproducing organisms? So we want to think about that question as we move through this lesson. It's a great idea. I know Jason got into the habit of writing down the essential question. That's a great idea to have, something that I think you all should consider doing because it really grounds you in what the goal of the lesson is. And it gives you an opportunity to go back at the end of the lesson and answer a simple question. Can I answer this question? Yes or no? If I don't know the answer to this question, then I didn't get what I was supposed to get from this lesson. And I might need to reach out to Mr. Rudd and say, hey, I need a little extra help. I missed something, or can you go back and go over some of these notes again? Whatever it might be, you want to be able to answer this question. And if you can, that's great. If not, you've got a little bit more work to do. So I'll leave this up for about 10 seconds so folks can capture whatever information they need to be in a good place to start this lesson. Okay. Let's start off with another quizzes review. Mitosis and meiosis are pretty much memorization heavy. You do just need to kind of memorize some things. You need to know prophase, metaphase, anaphase, telophase. You need to know some of the differences between these two processes. So a great way of doing that is playing games like this, quizzes, just to get your mind in a better position to memorize these concepts. This one is only 13 questions long. $725-135. I'm sending a link to the chat that will allow you to access this quizzes directly. Or you can go to joinmyquiz.com. And enter the game code 725135. I hear several of you joining already. Thanks, Michaela, Gloria, Rudy, and Monet. Excellent job. Let's get some more folks in here. We've got 15 people in our call today, so I want to see as many of us as possible in this game. Thank you, Kathy, Breon, Chandler, and Ayana as well. Let's get 7 more people, 7 more people. Thank you, Jason. We've got 9. We should have 6 more people who should be joining us. 6 more. Fraylin, are you able to join us? Neftali, are you able to join us? Rudy, Ayana, are you guys able? Oh, there's Ayana. She's already there. Sorry, Ayana. Rudy, are you able to join us? Thank you, Fraylin. Thank you, Cindy. Should have 4 more people. Okay. So those 4, you can join us as we start the game, but I'm going to go ahead and get started. Again, I want to see this accuracy up. Yesterday, we had an accuracy of about 60%. Let's see if we can get it even higher than 60%. I know this one might be a little bit harder. We've introduced some more concepts, but I want to see that accuracy up there, folks. Good job. Let's keep pushing it up. Come on. Uh-oh, it's falling down. Slow down. Take your time. I know you get more points for going fast, but make sure you understand the questions. Good. Good. Let's keep going. We're at 52. Can we get up past 60? We were at 60 yesterday. Excellent. 56. Monet is on a streak. Take your time, folks. Good job, Michaela. We're up there at 50. Come on. Come on. Let's go. Let's keep going up. Let's not go back down. All right, we're still at about 58. I want to see us get up there a little bit higher. Good job, though. Michaela's on a big streak, 7 questions in a row. Rudy's got a streak. He had a streak. Sorry, I ruined it, Rudy. Ayana's on a streak. $59. Let's keep going. Rudy is finished. Looks like Michaela is cementing her first place finish, but maybe Fraylin can catch her. Jason, you're in a good place too. Ayana, you're in a good place. I know some folks below the top 5 are also in good places. Michaela's rocking out. All right. Let's start to finish up. Let's take a few more minutes. I see everybody is starting to approach the end. 50. Yes, there it is. 60%. Come on. Can we stay up there? Can we hover around 60%? Good job, Jason. Well done. Well done. Michaela's finishing up. So is Ayana. Can you both get this last question right? I think you can. Ooh, I love it. 61%. I love it. $62%. Wonderful. Okay, we got 9 out of 12 folks done. Excellent, guys. We did well. We did really well with this. 9 out of 12 folks. So for those of you who are done, we're going to switch back to the notes. I know we've got 3 people who are still working. No big deal. Keep going. If you can finish, I know Cindy was removed from the game. No big deal. We'll come back and take a look at the questions a little bit later on, but I want to jump back into the notes. All right. So let me know. Do you guys have any ideas about this? On the left, we've got a set of fraternal twins, and on the right, we've got a set of identical twins. But you can see that the 2 young women on the left, they look really nothing alike, but it is true. So my question is, how is it possible that these 2 individuals share the same parents? And actually, they shared the same womb. They're actually twins. So what do you guys think? How is that possible? Why do they look so different?
I have no clue.
Thank you, Brianna, for acknowledging. Okay, so we don't know. Let's watch one of these videos. I love that Ayana is thinking about genes. That is the answer. They receive different genes, even though they came from the same 2 parents. So while they are very much alike, because they have the same 2 parents, they actually are no more alike than any other siblings. That's the thing with fraternal twins. They're basically just normal siblings who happen to be born at the same time. Identical twins are a different story because they are the result of the same egg. So they come from 2 different sperm cells, but the same egg. So that's a different story. But fraternal twins and triplets, they're basically no more alike than any other siblings. They just happen to have been born at the same time and in the same womb. So let's take a look at one of these videos that will kind of explain what we're talking about. This is it.
These pretty teens are best friends, and they're also something else. Something that may surprise you.
We're twins.
Believe it or not, they're twins.
Yep, we're twins. We're same
twin
mom,
sisters.
same dad.
You heard right. They have the same mom and dad. What are the odds that one would be black and one white?
A lot of people are just in shock.
Lucy and Maria Aylmer live in Gloucester, England. Maria has a caramel complexion, brown eyes, and thick curly black hair. Lucy has pale white skin, ginger red hair, blue eyes, and freckles. Most people have the same reaction when they hear they are twins.
They say, how did it happen? We don't believe you.
So, how did it happen? Their mom, Donna, is half black and half white. The twins' dad, Vince, is white. The couple was thrilled when a sonogram revealed they were expecting twins.
I just cried. It was shock.
Mom did a double take when she delivered one black and one white baby.
I'm so glad Mum dressed us in such cute little outfits.
Mom dressed her adorable twin daughters in matching outfits, just like twins everywhere. But by age 10, Lucy says she didn't feel like a twin, so why dress like one?
We don't look alike, so why should we have to wear the exact same thing?
Maria says she once wished for Lucy's straight hair.
I used to cry about it. I hate my curly hair.
And Lucy says she was sometimes taunted at school.
They thought I was adopted. You called me a ghost.
Now 18, the twins not only look different, they have very different personalities.
We're completely different.
Maria loves getting dressed up in chic clothing. She is very outgoing.
I love meeting people. Like, I'm not scared to approach people or anything, so.
Whereas I'm terrified of like going up to random strangers.
Lucy prefers casual clothes, but they've come to embrace their uniqueness as black and white twins.
If I have kids one day, they might come out looking like Lucy.
Okay. So that was interesting, right? Now, one thing we will discuss in greater detail in the future, and one thing that's still really important for me to say now, is that genes do not ultimately determine everything about a person. The reason for that is that your environment plays a role as well. The things that you're exposed to, the chemicals you're exposed to, the way that you're raised, the expectations that your parents and your teachers have of you, the expectations you place on yourself, the amount of sun that you get, all of these things contribute to changes in your physical characteristics, changes in your mental and emotional characteristics. And so there's always this intense interplay between your genes and the environment. Your genes play a large role in determining who you are, but so does the environment. And so in science, there is always this question of which one plays the larger role. And it's really a question that's impossible to answer. In some cases, we know, yes. Some women, for example, who have breast cancer definitely got that breast cancer because of a genetic predisposition to it. But at the same time, some women get breast cancer because of a poor diet or because they're exposed to some type of radiation. So there are different causes for all of these different outcomes. And it's oftentimes impossible to say if genes or the environment played the larger role. So when we look at these twins, yes, genetically they are different. And as Ayana pointed out, they're getting different genes from their parents. But at the same time, their environment has played a role in determining what their personalities are. We saw one twin who dressed a certain way and talked about being pretty shy and being horrified about approaching people. And the other twin saying that she was pretty outgoing. Their environments and the way that they were raised and the way that they like to be perceived by other people certainly plays a role in all of those things. And how they dress, how they present, how they choose to wear their hair, how they talk. We also see that race comes into play here. Now, race is not something that is rooted in biology. Skin color has really nothing to do with race, as we can see here. They have the exact same parents, but one girl probably would be considered black, and the other one probably would be considered white. Same parents, though. So race plays a role in how they are perceived. That would be more of an environmental factor than it would be a genetic factor. So these are good questions to ask yourself. We won't watch the second video for the sake of time, but it's basically the same story. As you can see, one twin. These are actually, I believe these girls were identical, but you can see that one of them has lighter skin and looks white. The other one has darker skin and looks brown. All right, so we just obviously did a review on the quizzes, but I want to make sure that you all have a good ability to distinguish between mitosis and meiosis. So what we've got here is a Venn diagram. Before we jump into this activity, I want you all to draw this Venn diagram. Make it really simple. You don't need to spend too much time doing it, but flip a page in your notebook and open up a new page and just draw big circles that overlap. And then write mitosis on the left and meiosis on the right. Jason, what I said was I just want you all to be ready to draw and reproduce this Venn diagram as we go through it. It's a good graphic organizer to have in your notes. It just helps you to see the differences between mitosis and meiosis. So the first thing I want you all to tell me, where does it go? Two daughter cells. Which of these processes produces two daughter cells? Is it mitosis or meiosis? Which one produces two daughter cells? Thank you, Ayana. Yeah, this one is mitosis. Next one. Which one undergoes two rounds of cell division? Would that be mitosis or meiosis? Which one undergoes two rounds? Excellent, Rudy. Thank you. This one is meiosis. So again, as we go through this, I really want to encourage you all to consider writing in your own notes this Venn diagram. Do yourself a favor. Okay, next one. Which one is a type of reproduction? Good, Brianna. That was a trick question. They are both types of reproduction. They are different types of reproduction, but they're both important types of reproduction. We'll get to the differences. Which one produces diploid or diploid cells? Yeah, thank you, Jason. Mitosis, that's right. I know what you mean. Mitosis, you are correct. It produces diploid cells. And a diploid cell, just as a reminder, has the full set of chromosomes. Full set. Okay, what about which one produces somatic cells? Which one produces somatic cells? Cells that are from your body. That's what soma means, body. So cells that make up your body. Which process produces those cells? Thank you, Jason and Brianna. That is a mitotic process to produce somatic cells. That's mitosis. Which one produces sperm and egg cells? Which process produces sperm and egg cells? No, not both of them. It is one or the other. Good, Rudy. And thank you, Ayana, as well. This happens via meiosis. Meiosis is responsible for producing our sex cells, the sperm and the egg cells. Only meiosis can do that. Now, what's another word for sex and, I'm sorry, for the sex cells? What's another word for it? It starts with a G. Wonderful. Thank you, Jason. Those are gametes. Gametes are sex cells, your sperm and your egg. All right. Which one is needed for sexual reproduction? This one should be pretty easy based on the last clue. Which one is needed for sexual reproduction? Wonderful. Thank you. Seems like you guys always type at the same time. Meiosis is needed for sexual reproduction. That's why it produces the sperm and the egg cells. Okay, which one is needed for asexual reproduction? Just give me the first three letters. You guys don't need to type out the whole word. Just give me MIT or MEI. Thank you, Brianna. One round of cell division. Which one is that? Thank you, Ayana. And which one produces haploid cells? Haploid. Good, Rudy. So mitosis produces diploid, meiosis produces haploid. Haploid cells have half the number of chromosomes. So in the case of humans, our body cells, our somatic cells have 46 chromosomes, but our gametes only have 23. They have half the number, okay? And that's why meiosis produces those sex cells. It's needed to get through or get to that half number. Which one produces unique daughter cells? Thank you, Ayana. Yeah, meiosis produces totally unique daughter cells. Every single sperm and every single egg is different than one another, than all the others. Lastly, which one produces identical daughter cells? This should be a quick one. Wonderful. Yeah. So take a look at this Venn diagram now. This is something that you should have in your notes. Graphic organizers like this are great, guys. They really, really help you to understand and to remember facts about similar but different things. In this case, similar but different processes. They're both needed for reproduction, but different types of reproduction, different results, different types of division. Leaving this up for about 10 more seconds for folks to take a picture, a screenshot, or finish writing. Okay. So today's lesson is again about genetic variation. Obviously, it's already 11 o'clock, so we're starting to bleed into the asynchronous time. I didn't upload a new assignment for today because I see so many of you have a lot of missing assignments that you need to get rid of or get done with. You need to do them. That's the best way I can say that. A lot of missing assignments that still need to be done. So I didn't want to give any more work over the weekend and give you all an opportunity just to catch up. I know some of you have submitted some late assignments. I do plan on grading those this weekend, but just be patient with me. I typically only like to grade at the end of units. So I'm going to get that done this weekend, but just be patient. I'm also going to upload your test scores. And I know that there are a few of you who have not taken that Unit 2 test and a couple of you who didn't take the Unit 1 test. Let's see. I don't think those people are on this call right now, but yeah. Chandler, you and I still have to find a time to meet to take that Unit 2 test. But I do appreciate you reaching out and trying to schedule an appointment. So we'll get that done, but you guys will see your grades change primarily because of that Unit 2 test. But use the weekend to make sure that you get these missing assignments submitted if you know you do have missing assignments. If your question is, how do I know if I have missing assignments? PowerSchool will tell you, and so will Canvas. On PowerSchool, you'll see that your assignments that are marked with an orange square, that means that it's missing, that assignment was not submitted. Now, if you go in and you're like, I'm pretty sure I did submit that, then just wait and see until after this weekend. Like I said, I will be changing grades for people who have submitted late assignments. But Canvas will also tell you which assignments you have not done, so you can go in that way and see what you're missing. Please focus on the asynchronous assignments because they're worth more points. The warm-ups and exit tickets are important for all the reasons that I've talked about already. but the asynchronous assignments are basically the second half of the class. You can't really skip over that. You've got to make sure that you're doing those asynchronous assignments. So that being said, I probably will use the rest of this time to lecture. We've only got 30 minutes left. We might have a little bit of time left at the end to do the exit ticket, but for the most part, I just wanted to let you guys know that this weekend, you won't have any new assignments. It's just going to be an opportunity for you to get caught up if you know you're missing something. Okay. Of course, you can always reach out to me if you have questions. Reach out via Remind, text message, email, Canvas, however you want to get in touch with me. That works. So, meiosis and genetic variation. What is genetic variation? Well, genetic variation is the diversity of gene frequencies between individuals or between populations caused by several different sources. We're going to talk about 5 of those sources today. Remember, the words that are highlighted in yellow are words that I really encourage you to write down in your notes. So genetic variation is the diversity of gene frequencies, DNA frequencies. And it results in families that look like the family on the right. Those puppies are from the same litter, but we can see they've got some differences. Some of them have larger eyes than others. Some of them have darker coats than others. Some of them have straighter hair than others. And we can see their parents sitting up as well. Okay. So the number one source of genetic diversity, the number one primary source of genetic variation is mutations. I'm sure you all have heard of mutations before. You probably have heard it in a sci-fi context where you hear about someone who gets mutated and they become some type of organism with special powers, whatever. But mutations happen all the time, and they're happening all the time in each of our bodies. Our cells are constantly being mutated. Oftentimes, the mutations are ineffectual, and I'll talk more about that. But a mutation is defined as a change in the structure of a gene caused by the deletion, insertion, or substitution of a nucleotide base. So think back to Unit 1 when we first talked about nucleic acids. We first talked about DNA. I told you guys then that there are 3 parts of a nucleotide. One of those parts is called the nitrogen base. And when we're talking about DNA, we've got 4 different bases, A, T, C, and G. We'll talk more about mutations and we'll talk more about these bases A, T, C, and G later on in the course. But what you need to know now is that mutations can be inherited. So when your DNA gets mutated, let's say that you spend too much time in the sun and the UV rays mutate some of your cells. Some of those mutations can actually be passed on to your children. So mutations can be inherited, meaning they can be passed on from one generation to the next. Oftentimes, we assume that mutations are a bad thing, and that can be true. Mutations can be detrimental. They can be bad things. But mutations can also be beneficial, meaning that they can help an organism to survive. Sometimes mutations give an organism a better chance at survival, or they give an organism a better chance at having children. And these are beneficial mutations. We can think of the classic example of giraffes. Giraffes, many hundreds of thousands of years ago, were basically just horses. They were tall horses. But over time, some of them ended up getting mutations that made their necks longer and longer and longer. And this was beneficial to the giraffes because it gave them access to food that other organisms couldn't reach from the ground. They had these long necks that gave them, it put their mouths 20 feet in the air. And so they could have access to these tall branches that other organisms simply could not reach because they were too short. So that's an example of a beneficial mutation. The vast majority of mutations are what we call ineffectual. That means that they don't have any effect. They're neither good nor bad. So oftentimes those mutations that are happening in your body, they end up doing nothing. They're there, they're just a mistake. The cell might go back and correct them. That cell might just die, and nothing ends up happening to you. Nothing good nor bad. Nothing is passed on to your children. Are there any questions about this concept of mutations? Okay. So there are different types of mutations, and again, we'll talk more about that. So there's nothing for you all to write down here, but I'm just going to quickly run through a few of these types of mutations. What we have here is called a point mutation. So on the top, we can see the original sequence of DNA. We see those bases. We see T, then A again, then C, then T, then G, then C, then A, then G, then G, then T. In the human genome, we've got about 3 billion of those. So 3 billion letters like that. Same for T, A, C, G. In this section of DNA, we see that there's a C where there should be a T. That's called a point mutation, meaning only one of these bases is changed. Often these mutations are ineffectual. They don't have a positive effect. They don't have a negative effect. They're just there, oftentimes. Sometimes they can be detrimental. Sometimes they can be beneficial, but more often than not, nothing happens. They're so small that they often don't have an effect on the protein that's made. We have to remember that these genes are the instruction manuals to make a protein. Or in my case, I like to think of them as the recipe books to make meals. So think about it. If we had a recipe book that was, 20,000 recipes in it, and there were 3 billion letters in this recipe book. If we only changed one letter, it's probably not going to have a big effect. It might be, changing the I in chicken to an A. So now it says chacken. We basically still know what that says. Yes, it's not a real word. Yes, it might be a little confusing at first. What do you mean I'm supposed to grill chacken? But at the end of the day, we can still kind of come to the conclusion, oh, that's probably supposed to be an I. It probably should say chicken. So in the grand scheme of things, it's not actually having a big effect on the meal that we make. Then there are what we call substitution mutations. A substitution mutation is going to take a group of bases and switch it out. So as you can see, we're supposed to have a TGC here, but it's replaced by a GAT. That more often than not is going to have an effect. We can't say if it's a positive effect, but it could also be a negative effect. We can't say for sure, but it will probably have an effect on the protein that is made. Because this is a larger change. Instead of just one base, now we're changing 3 bases. A group of 3 bases is called a codon. We'll talk more about that later in the course. But if we change this codon, we're substituting a codon, that's more likely to have an effect on the protein that's made. So back in my example, now instead of just changing the I in chicken, maybe I change the CHI. I'm changing 3 letters. And I change it from CHI to BRA, bracken, grilled bracken. Now we have no idea. We're like, what is a grilled bracken? I've never heard of that. Is that some type of exotic fish? Is that some type of rare red meat? We don't know. Okay, so changing 3 letters is more likely to have an effect. Maybe it has a good effect. Maybe I do find that there's some exotic fish called the bracken that's really good when grilled. Maybe that ends up being a good thing. But maybe it has a negative effect. Maybe it's so bad that we don't even know what it is, and so we just kind of delete that gene. That's going to be a bad thing. Then also there are deletions. When we delete even just one base, what ends up happening is that everything that comes after that deletion is now messed up. The reason for this is that we read codons in groups of 3. So here I should be reading TAA, that's one codon, then CTG, that's another codon, then CAG, that's another codon. But now because of this T being deleted, I now have TAA, so that first codon is still good. It's still exactly what it should be. But instead of CTG now, I have CGC. That's messed up. And everything else after this deletion is now messed up because now I have AGG instead of having CAG. So these often have they have deleterious effects. They have negative effects. They often ruin the protein that's supposed to be made. We'll talk more about these mutations and get some good practice with them later on in the course, but I just wanted to introduce you all to them quickly. The second source of genetic variation is called crossing over. Not a crossover for my hoopers. It's called crossing over. Crossing over is the exchange of genes between homologous chromosomes, resulting in a combination of parental traits. That sounds like a mouthful, but the image at the bottom really demonstrates what we're talking about. We've got these homologous chromosomes. One is pink, one is purple. We can see that they exchange some of their genetic material. So now, let's say that the pink one is coming from the father. The father's chromosome has a little bit of genetic information from the mother. And the purple one is coming from the mother. So the mother's chromosome has a little bit of genetic information coming from the father. And this crossover greatly increases genetic variation. And it happens every time, only during prophase 1 of meiosis 1. Crossing over only happens during prophase 1. Crossing over is a process that does not happen during mitosis. Remember, mitosis produces identical daughter cells. So mitosis is not expected to give us any genetic variation. We're just making copies of the parent cell. We don't want genetic variation then. Meiosis is when we do want genetic variation, and this is one of the ways that we get it. We can see in this GIF, or GIF, however you want to say it, we'll call it an animation. We can see this process of crossing over happen. Some of the red chromosome gets transferred to the blue chromosome, and some of the blue chromosome is transferred to the red chromosome. This process is also known as recombination. Recombination. Let's watch it one more time. There's the crossover happening. All right. The third source of genetic variation is independent assortment. Independent assortment is a law that states that different genes are inherited independently of one another. Basically, this means that genes are not linked. So inheriting one gene from a parent does not increase the chances of inheriting another gene from that parent. Just because you got your eye color from your dad doesn't mean that you also got your eyelash length from your dad. just because you got your hair color from your mom doesn't mean that you got your mom's hair texture too. It's totally random which genes you get from which parent because genes are not linked. So this is why some people look just like their father, but then they have a sibling who looks just like their mother. And then another sibling who might not look anything like either parent. And another sibling who looks like a grandfather. This is a random process. Genes are not linked to one another. So just because you've got the genes for your mom's height doesn't mean that you also have the genes for your mom's physique. And if we look at this image on the right side of our screen, we see a demonstration of this. Genes are not linked to one another. So this means that there are many, many combinations of different genes that we can produce. Every single gamete is going to be slightly different because they're getting different alleles from each parent. And an allele is just a version of a gene. So some sperm are going to have the allele for dark hair color. Some sperm are going to have the allele for brown hair color. Some sperm are going to have the allele for, in some cases, even blonde hair color. Just depends on what genes your dad has. Same thing with the eggs. Depends on what genes your mom has. But it's a total, you've got all these different possible combinations. That leads directly into another one that we'll talk about next. But before we do that, we have to talk about nondisjunction. Hold on. Yeah. Nondisjunction is the failure of homologous chromosomes or sister chromatids to fully separate during meiosis. That's what nondisjunction means. So disjunction means to separate. Nondisjunction means failure to separate. This results in some of the gametes, some of the sex cells, having more or less genetic information than they really should. And if those gametes end up fertilizing, so let's say we've got a sperm cell that is altered because of nondisjunction. If that sperm cell ends up fertilizing an egg, then the resulting offspring is going to have an odd number of chromosomes. Instead of having 46, they might have 47. Instead of having 46, they might have 45. They have an odd number. So nondisjunction is the failure of homologous chromosomes or sister chromatids to separate during meiosis. So here's normal meiosis. We've talked about this before. This cell has already gone through interphase. This is the parent cell. It's already copied its DNA. In meiosis 1, we can see that these X-looking chromosomes, these homologous chromosomes, are supposed to be separated from one another. And then in meiosis 2, we actually rip apart the chromosomes. So the sister chromatids are separated. That's normal. So this is what we should normally end up with. Every single gamete has N number of chromosomes, If the non-disjunction happens during meiosis 1, as we can see here, one of the cells is going to end up with two of these gametes are going to end up with too many chromosomes. They've got too much genetic information. Two of them end up with not enough, because this chromosome should have ended up over here, but instead it got pulled to this cell. It didn't separate how it was supposed to. If the non-disjunction happens in meiosis 2, everything is fine in meiosis 1. We separate the homologous chromosomes properly. But in meiosis 2, we end up not separating one of the chromatids. So now one of the gametes has too much, one of them has too little, and two of them are normal. So you see what the results of non-disjunction are. Some of the gametes are going to end up with too much, and some of them end up with too little. Exactly how many of them are altered just depends on when the non-disjunction happens. If it happens early, then all four of the resulting gametes are altered. If it happens during meiosis 2, then only two of the resulting gametes are altered and two of them are normal. What are the practical implications of this? Well, the practical implications can be pretty severe. What we're looking at here is a karyotype. A karyotype is just a 3D representation of a person's chromosomes. So we can see the 23 chromosome pairs here. Here's chromosome pair 1, chromosome pair 2, 3, 4, so on and so forth. As you can see, as we go, they get smaller and smaller and smaller. What you should notice is that as we get to pair number 21, we actually don't have a pair. We've got 3 chromosomes there. So non-disjunction resulted in this person having 3 chromosomes in the 21st position, where they should have had only 2. This results in a condition that's called trisomy 21, but more commonly we know trisomy 21 as Down syndrome. So Down syndrome is the result of non-disjunction, leading to a person having 3 chromosomes in their 21st position where they should only have 2. Now, obviously, people with Down syndrome, are often able to live a semi-regular, semi-normal life. Unfortunately, their lifespan is typically shorter than people who do not have Down syndrome. Oftentimes, they're unable to reproduce because of this condition. Oftentimes, they do have some cognitive impairments. And often they do have similar physical characteristics. They're a little bit shorter. They're more likely to be slightly overweight. But these things are not guaranteed, right? There are people who have Down syndrome who are able to live, they're able to be actors and athletes. They have different physiques. They have different cognitive abilities. So it's not guaranteed that Down syndrome will result in extreme impairment. And then the last source of genetic variation, is fertilization. Fertilization is the process by which a sperm and egg cell unite to create a zygote. So a zygote is that very first cell that eventually becomes a full-grown organism. It's the very first cell. And that zygote, as soon as it's created, it starts undergoing mitosis and making copies of itself. And then some of those cells will eventually become muscle cells. Some will eventually become skeletal cells, bone cells. Some will become red blood cells. Some will become nerve cells. That's a process called gene differentiation that we'll talk about later on. But this zygote is that original OG cell. As soon as the sperm and the egg unite, they create a zygote. And this is how we see a unique combination of genes. Some of those genes are from the mom. Some of those genes are from the dad. Every time it's totally unique. All right. So let's just quickly go over a few of these quiz questions. 11 of the 12 of you finished. I think Cindy got kicked out because of technical errors, not on her part. Something went wrong with the quiz. So let's look at, we had a tough time with number 3. Number 3 asked, the result of meiosis is. Okay. So hopefully we can clarify this now, or hopefully you all feel better about this now. 7 of you got it wrong when we took the quizzes, but based on the Venn diagram that we just did, hopefully you all feel better answering this question. What is the answer here? Would it be 2 identical daughter cells, 2 different daughter cells, 4 identical daughter cells, or 4 different daughter cells? Remember that Venn diagram. What does meiosis produce? 2 or 4, identical or unique? Wonderful. Thank you, Ayana. 4 different daughter cells. 4 different daughter cells. Meiosis will always produce 4 sperm or 4 eggs, and each one of them is different from the others. Okay. The other one we struggled with a lot is number 11. Only 2 people got this right. Okay. It asks, what is the correct order of the phases of mitosis? Can't click on the image. Hold on. End it. Then hopefully I can click on it. Fraylin came in 3rd, Jason in 2nd, and Michaela rocked out, got 1st place. Excellent job. Questions. All right, what is the correct order of the phases of mitosis? I'm going to assume that you all struggled with this one because the picture is so small. Even I can't click on it right now. But when we look at these images, we've got to be able to tell what exactly is going on. Typically, we will start with, let's see, hold on. Yeah. All right. So they kind of probably threw you guys off a little bit here because they put in an image between prophase and metaphase. There is a phase it's called prometaphase because it comes between prophase and metaphase. And that is represented by picture F here. And during prometaphase, all that's happening is that the chromosomes are actually moving towards the center of the cell. All right, so A is the first image. A represents prophase. We can see the centrioles and the spindle fibers starting to form. Then we move over to F. F is as the chromosomes are starting to move towards the center of the cell. Then C, which is metaphase, as they actually line up in the middle of the cell. So I can see why so many of you chose these 2 options because C is in the 3rd place. After metaphase comes anaphase. So that has to be E, as we're seeing the chromosomes be pulled apart from one another into sister chromatids. Then after anaphase comes telophase. I can also see why this one was difficult because there's not really a big difference between A and D. So I trust that you guys understand how to identify these different steps, these different phases of mitosis. That one was a little more difficult. But we'll leave it there. Good job today, folks. Like I said, I will grade and I'll let you know if there are any assignments that I'd like you to do, but I do want you to focus on missing assignments. Almost everyone in this class has at least one missing assignment, even if it's as small as a warm-up. So please take time to do that this weekend, but also take time to relax, rejuvenate yourselves. We've got a lot of 5-day weeks, unfortunately, back to back to back to back. So we've got a stretch of time in which we're going to need to be focused and ready to go. So enjoy your weekend, be productive, but also be kind to yourselves and kind to your family members. I'll talk to you guys on Monday.